This is our second podcast in our series on epithelium. And in the first podcast, we finished with talking about epithelial cell polarity when we were talking about structures on the apical domain of epithelial cells. I'd like to now look at structures associated with the lateral domain of epithelial cells. And basically here we're looking at structures on epithelial cells that allow contact and communication between adjacent cells, and this usually occurs with specialized attachments or junctional complexes. Now, the lateral domain in epithelial cells is specialized for cell-to-cell adhesion with cell adhesion molecules that actually form parts of junctional complexes. And we're going to be looking at several types of junctions. First, we'll be looking at occluding junctions, also called tight junctions or zonula occludens. These structures form impermeable intracellular diffusion barriers between cells. They maintain physiochemical separation of tissue compartments, and they maintain distinct plasma membrane domains, especially in epithelial cells. We'll next talk about anchoring junctions, either zonula adherens or macular adherens. These junctions provide mechanical stability between cells by linking the cytoskeleton of one cell to the cytoskeleton of another cell, or, as we'll see in a specialized case, linking the cytoskeleton of cells to the basal lamina. These junctions are particularly important in maintaining the structural integrity and structural unity of epithelium. These junctions also play some roles in signal transduction, in cell-to-cell recognition, especially during morphogenesis and differentiation as embryos develop. And finally, we'll look at communicating junctions, also called gap junctions. These structures allow for chemical and electrical coupling between cells, and they play an important role in the coordination of cellular activities for homeostasis. I've got an electron micrograph displayed on the left-hand side of this image, and it's showing zonula occludens, zonula adherens, and then macula adherens. So first, let me just define something and just say that a zonula is a belt-like structure that encircles the apical portion of the cell. So you'd have a zonula occludens wrapping around the apical portion of cells, this being one cell or a portion of one cell, this being a portion of another cell. So the zonula occludens wrapping around the apical boundaries of these cells, the zonula adherens just below the zonula occludens, but also wrapping around the apical boundaries of cells. So zonula, a belt-like structure that encircles the apical portion of the cell, macula, a spot or button-like structures. So now in this diagram there are portions of 1, 2, 3 cells, and the artist has drawn these with microvilli on them, so we might think of these as like columnar epithelial cells in the GI tract, just as a hypothetical cell. And the artist has separated the microvilli on these epithelial cells, and you can see in this region, these would be the actin cores shown here, or shown here in the microvilli, the actin cores going into the microvilli and interacting with substances in the cortical cytoskeleton. But then the artist has now drawn these two zonulas, with the zonula occludens on the more apical region of the cells, and then the zonula adherens also on the apical region of the cell, but below the zonula occludens. Then the artist has drawn macula adherens, also given the terms desmosomes, throughout the cells, and we will talk a little bit later about hemi-desmosomes and focal adhesions, other types of junctional complexes on these cells. So here is just a simple-minded schematic diagram showing the specialized junctional complexes between epithelial cells. The apical boundary of the cell shown with these microvilli would be drawn, so you could imagine a portion of one cell, another cell, and a portion of a third cell. The zonula occludens most apical, then the zonula adherens, macula adherens, gap junctions, and then hemi-desmosomes. Now, another important point the artist makes here, the zonula adherens interacts with actin cytoskeletal filaments. So the adherens junctions interact with actin. The macula adherens or desmosomes interact with intermediate filament cytoskeletal components, most notably keratins. These hemi-desmosomes also interact with keratin intermediate filament proteins, and we'll talk about that in a few minutes. A little more detail on these. The zonula occludens or tight junction functions to seal neighboring cells together in an epithelial sheet to prevent leakage of molecules between cells. The tight junctions also function to maintain very distinct plasma membrane domains. So two functions of tight junctions: to maintain distinct plasma membrane domains and to prevent leakage of material between cells. The adherens junctions join an actin bundle in one cell to a similar bundle in a neighboring cell. So these junctions are actually binding the actin cytoskeletons from one cell together to the actin cytoskeleton in another cell. So these junctions provide very strong structural integrity between cells. The adhering junctions, zonula adherens, are also known as belt desmosomes. On the other hand, the macula adherens or spot desmosome joins intermediate filaments in one cell to those in a neighboring cell. The intermediate filaments in these types of junctions would be keratin or various cytokeratins. Gap junctions allow passage of small molecules, usually water-soluble ions and very small molecules, from one cell to another, and as we suggested just a couple of minutes ago, they allow cells to be in communication and to help coordinate functions in homeostasis. And we'll talk about hemi-desmosomes. These are junctions in a sense that they're structures. They're not really junctions because they don't bind cell to cell, but they bind cell to the basal lamina. So they function by attaching intermediate filament proteins through the membrane onto proteins in the basal lamina. Here is a light micrograph of some epithelial cells in the GI tract. Here where I've got my pointer, that would be the luminal surface of these cells. This would be like the lumen of the GI tract. So here would be an apical domain, and here would be an apical domain on the cells on the opposite side of the lumen. The electron micrograph shown on the right would be an electron micrograph through this region of these cells, and the micrograph shows the zonula occludens, the zonula adherens, and a macula adherens. What's notable in this micrograph, you can see portions of the microvilli. You can see filaments that are going into the microvilli. These would be actin filaments. These would also be actin filaments associated with the zonula adherens. These filaments on the macula adherens would be keratin filaments. Let's talk a little bit about the tight junction. Here I've shown the apical boundary of the cells. So down here would be the luminal pole of these epithelial cells. Here is the lumen in the light micrograph, and so I would be looking in the diagram at a region like this. So the zonula occludens, diagrammed here, is found at the apical or luminal pole of the epithelial cells. Provides some structure to the membranes. Integral membrane proteins of adjacent cells actually fuse. The zonula occludens forms a seal between adjacent cells, separating the luminal and intracellular spaces. So, for example, if this is luminal space would be in here. This would be intracellular space here, and intracellular space here, because you would imagine this would be a cell like this. This would be the portion of another cell or the portion of a third cell. These zonular occludens or tight junctions are forming seals. They're separating the luminal space from the intracellular space. That means that material doesn't flow between the cells, but would have to cross these tight junctions. The number of rows of these where these proteins fuse in the tight junctions determines the tightness of the seal, and that will become clear, I think, in a couple of minutes. Tight junctions maintain apical and basolateral plasma membrane domains. Remember that proteins can move freely in this lipid bilayer because this is essentially a two-dimensional fluid, and the proteins can float freely in this two-dimensional fluid, except that proteins in this apical domain cannot float past the tight junctions. And likewise, proteins in this basal here or lateral domains, proteins can float freely between these domains, but they can't pass these tight junctions to get into the apical domain. So the tight junctions are maintaining very distinct plasma membrane domains. These tight junctions depend on calcium. They'll form as they're needed, but they don't require protein synthesis. And this is an electron micrograph, a freeze fracture basically through tight junctions. Here is a light micrograph of the area that's being fractioned. So here on the light micrograph is the luminal space, right where my pointer is. So the luminal space is down here. Here are the microvilli fractioned. Here is the row of tight junctions. You can see these various ridges. These would be the seals of proteins that are forming and actually fusing from membranes one cell to another. Here in the transmission electron micrograph, you can see regions where the plasma membranes, one cell here, another cell here, the plasma membranes have actually fused, as shown here. Here there's a space between the plasma membranes, here fused again, here space, here fused again, here space. So if I were to go over to on this side of the micrograph, the ridges here, or these rows, represent the fusional portions of the junctions, and these spaces here would represent the spaces that you see here on a transmission micrograph. It's almost like the tight junctions are the stitches on a bed quilt. You can think of each side of the quilt as membranes of adjacent cells, and the stitching between the quilts of the tight junctions holding those cells together so that material doesn't pass around that stitching, if you want. There are three major groups of transmembrane proteins that are associated with the zonular occludens. Let's look at this cartoon just for a second. Here is one plasma membrane, here is another plasma membrane, and here the integral membrane proteins from one membrane and another membrane. They're actually causing the membranes to fuse, to come together. So the major types of proteins, there are proteins called occludens, and occludens maintain the barrier between adjacent epithelial cells. They bind to cytoskeletal or scaffold proteins within the cell. They maintain the barrier between apical and lateral plasma membrane domains. Claudins maintain seals between adjacent cells. They can form aqueous channels for paracellular transport of ions and small molecules, and we'll come back and tell you what paracellular transport means in a few minutes, and we'll contrast that to transcellular transport in a few minutes. And then finally, there are these JAM proteins, or junctional adhesion molecules, that actually associate with the various Claudin proteins. We've seen a version of this slide in a previous presentation. We could say this is representing the GI epithelium in the gut. Here is the luminal space, the space of the epithelial cells, and then the extracellular space on the basal surface of these membranes. High glucose concentration in the cell. Remember, there's glucose that's in the lumen as you take in food. This glucose has to cross the plasma membrane going against a concentration gradient. It's taking a ride on a sodium-driven glucose importer, and then there are these glucose transporters shown in red on the lateral and basal surfaces of the epithelial cells that simply transport glucose down its concentration gradient so that it gets to this intracellular space and eventually into the blood capillaries. Now, I'm going to try to simplify this slide as we go. So what's colored on the top in orange is the lumen of the gut. It's the apical domain. What's colored in blue is the basal domain, and so you see the basal lateral domain. So what's in blue is extracellular space. The tight junctions are forming a seal between the luminal extracellular space and the extracellular space upon which these cells rest. I think you can imagine any of these proteins could float freely in the apical domain of the membrane, but they cannot pass these tight junctions. Any of these proteins can float freely in the basolateral domain of the membrane, but they cannot pass these tight junctions. Now that makes perfect sense physiologically, because here the sodium-driven glucose imports you're going to want to have in this apical domain of the membrane. Here, these passive glucose carriers you're going to want to have in the basolateral domain. What does paracellular movement mean? Paracellular movement means movement of material between cells through a tight junction. Normally, you have very little paracellular transport between these tight junctions because of Claudin seals that prevent movement through these tight junctions. If there is movement through these tight junctions, one would call them leaky tight junctions, and that's why we make the comment that the number of rows of tight junctions that are designated by these black structures, the number of rows of tight junctions would determine how tight the water tight seal is between cells. That's paracellular movement then between cells, as opposed to transcellular movement across the cell itself. So we would say in this example, glucose is not moving in a paracellular manner through the cells. Rather, it's moving across the cells, kind of by transcytosis, getting across the membrane here and then across the membrane this way. So transcellular movement versus the paracellular movement between cells. Now, lest you get confused, a tight junction doesn't mean that it's holding the cells together tightly. That's a little bit counterintuitive. It's not providing structural integrity of the epithelium. It's a tight junction in that it's maintaining plasma membrane domains, not letting material move from the apical domain to the basolateral domain, and likewise not letting material move between the cells. That's why it's called a tight junction. There are a couple of interesting clinical correlations with tight junctions and tight junction proteins, because some of these proteins turn out to be the targets for various pathogens. So, for example, Clostridium perfringens, the microorganism that causes food poisoning, secretes an enterotoxin that binds to Claudin. That enterotoxin forms pores in the apical domain of the GI epithelium, and you get massive dehydration due to fluid loss in the paracellular movement. This enterotoxin makes the tight junctions very leaky. So you imagine a massive fluid loss, essentially a massive diarrhea. The microorganism that causes gastric ulcers, Helicobacter pylori, proteins target Zonula, Claudin-1, and various junctional adhesion molecule proteins. This disrupts tyrosine kinase signaling and disrupts the cytoskeleton of these cells. And finally, the common household dust mite, and I'm not even going to attempt to pronounce the scientific name, but there are peptidases that are in the fecal pellets of these organisms that when you inhale the fecal pellets with dust, the peptidases break down the zonula occludens in respiratory epithelium, and that makes individuals susceptible to a variety of inhaled allergens. So some of these tight junction proteins do have important clinical implications if they are targeted by various pathogenic proteins from pathogenic organisms. Say a few words about anchoring junctions or adherens junctions. Anchoring junctions provide lateral adhesions between epithelial cells. Integral proteins in one membrane link to cytoskeletal proteins, and the integral proteins in one membrane link to the integral proteins in another membrane, so you're essentially binding cytoskeleton to cytoskeleton from one cell to another. By virtue of these integral membrane proteins, these adhering or anchoring junctions contribute to the structural integrity of epithelium. There are various cell adhesion molecules, which are transmembrane proteins that are found on the extracellular domains of the membranes. The intracellular domains of these cell adhesion molecules bind onto the cytoskeleton in cells. Heterophilic binding would be binding of different types of cell adhesion molecules from one cell to another. So one cell adhesion molecule in this cell binds onto a different cell adhesion molecule in that cell. That's heterophilic binding as opposed to homophilic binding. The same type of cell adhesion molecule in one cell binds the same type of cell adhesion molecule on the adjacent cell. The point is that this binding of cell adhesion molecules on adjacent cells provides fairly substantial interactions between the epithelial cells and contributes to the integrity of the epithelial sheet of cells. Now, a zonula adherens is a belt junction around the apical portion of cells. These zonular adherens proteins interact with actin in the cortical cytoskeleton. And then we'll talk about macular adherens or desmosomes. These are called spot junctions. These structures interact with intermediate filaments, keratins. And we'll look at each of those in turn. Here is a table that shows the various cell adhesion molecules associated with anchoring junctions. I put this up just for informational purposes. We can talk about classical cadherens or desmosomal cadherens, several varieties. There's epithelial, nerve, placental, and vascular endothelial cell cadherens. These proteins are very much dependent on calcium and magnesium. They're usually homophilic binding. The cadherens interact with actin filaments. The desmosomal cadherens, like desmoglein, interact with intermediate filaments. The classical cadherens are in the adherens junctions, the desmosomal cadherens, as you might expect in desmosomes. And then there are various types of selectins that we'll talk about later in the course, but there are selectins like L-selectins for leukocyte selectins, epithelial selectins, placental selectins. These selectins are very important in cell migration and what we call diapedesis of, for example, leukocytes across the walls of blood vessels. Various integrins bind cell to extracellular matrix. So, take-home message from the table: the classical cadherens and selectins bind cell to cell, and then integrins bind cell to extracellular matrix. So cadherens, they're calcium dependent. They're usually single pass integral membrane glycoproteins, so the sugar residues are on the extracellular domains of the proteins. These bind cell to cell, so you find them in zonula adherens, you find them in macular adherens or desmosomes, and you'll find them in independent focal contacts within cells. Intracellular domains of these cadherens bind to actin or keratin components of the cytoskeleton. cell adhesion molecules in the immunoglobulin family are calcium independent. Most notable of these are the neural cell adhesion molecules. These are single pass integral membrane glycoproteins. They have repeating units that are similar in structure to immunoglobulins. The higher the Ig-like or immunoglobulin-like content to these cell adhesion molecules, the stronger the binding. They also have repeating sialic acid residues, and the higher the sialic acid component in these proteins, the weaker the binding. These cell adhesion molecules cause cell-to-cell binding, but they cause weak binding, so they're just binding cell membrane to cell membrane. There's no cytoskeletal interaction, so this is a weaker type of interaction, as opposed to the cadherins, which, because the cadherins are binding the cell membranes to the cytoskeleton, a much stronger type of interaction. And then integrins, these are calcium and magnesium dependent. These are single pass heterodynamic integral membrane glycoproteins. These are present in focal adhesions. They bind to the actin cytoskeleton. They're present in hemi-desmosomes, and in this case bind to the keratin cytoskeleton. The integrins usually are receptors for extracellular matrix proteins, so they usually bind the cell to its extracellular matrix. So these integrins commonly have receptors for molecules like collagens, fibronectins, and laminins, which you know of as extracellular proteins in connective tissue. And then the cell adhesion molecules in the Selectin class, these are calcium dependent. They're single pass lecithin containing integral membrane proteins. They bind cell to cell by specific carbohydrate residues. They can attach to the actin cytoskeleton. The P-selectins are particularly involved in implantation of embryos, and then the E and L selectins are involved in chemotaxis, diapedesis, and in wound repair. And we'll show examples of this later in the course. Now, let me talk a little bit about the zonula adherens anchoring junction. Plasma membranes of adjacent cells approximate each other, but they don't actually touch. So in the cartoon, you can see the tight junction of zonula occludens, where the plasma membranes actually touch and fuse. Here in the zonular adherens, remember zonular adherens has a belt that goes around the apical surface of the cell just below the zonula occludens. The cell membranes approximate each other, but they don't actually touch. There's going to actually be a space between the cell membranes. The cadherin proteins bind to actin of the cortical cytoskeleton, the so-called terminal web, and externally to other cadherins to hold epithelial cells together. So these junctions are calcium dependent. As we said, they form a belt-like structure just below the tight junctions, so they circumscribe the apical region of the cell. These zonular adherens are also given the name belt desmosome. We've already alluded to this earlier. The terminal web provides kind of a structure to the apical region of cells. They allow anchoring sites for actin filaments that go up into the core of microvilli. Just as an aside, it's not something that's critical for our understanding right now, but it does have some important physiologic significance. Microvilli, they're not motile like cilia would be, but these microvilli can get shorter or they can get a little bit longer as contraction of the actin cytoskeleton might alter their shape a little bit. So you might imagine these microvilli might scrunch down if these actin components get a little bit shorter. Likewise, they might, as the actin components relax a little bit, these microvilli might get a little bit longer, and so you might imagine that would provide a little more membrane surface area. Here is a diagrammatic view of the zonula adherens, so it's showing cytoskeletal components like actin molecules in adjacent cells. So this would be a region of one cell. This would be a region of another cell. Here are the actin molecules shown in green, and various other cytoskeletal components like alpha-actinin and vinculin. But then here are the cadherin molecules, these transmembrane integral proteins spanning the membrane, and they're binding onto cadherin molecules in an adjacent cell. So you can imagine that these two membranes of cells are coming fairly close together, but there's an interaction, if you want, of the cytoskeleton of one cell to another cell because of the binding of the cadherin proteins from one cell to another. As shown in the electron micrograph, there is actually a space between the cells, but you also get a sense that there's some type of a proteinaceous material between the cells, and that's what's reflected here in the diagram. And then here you can get a sense that this is representing, on the diagram, the arrows on the electron micrograph are reflecting the cytoskeletal components that are drawn on the diagram. So again, you can imagine these zonal adherens holding the cytoskeletons of one cell tightly to the cytoskeleton of another cell, this helping to maintain the integrity of the epithelial sheet. So for example, these cells would have high resistance to, you might imagine, shear forces. Desmosomes, or macular adherens, diagrammatically shown here, interacting with keratin in the cytoskeleton. They have very characteristic electron-dense protein plaques, and then these intracellular proteins between the cells like desmogliin, desmocolin, that are integral membrane proteins spanning the membrane, binding to similar integral membrane proteins on an adjacent cell. So the membranes themselves don't fuse, but the integral proteins bind. So there's still actually a space between these membranes. These macular adherens form button-like or spot wells that bind adjacent cells together. They're calcium dependent. These are also very common in cells that are subject to lateral shear forces. So you find them in the epidermis and the skin. You find them particularly in the epithelium of uterus. You also find a specialized type of macular adherens in cardiac muscle, and we'll talk about that later in the course. Cadherins bind to keratin filaments of the cytoskeleton, and they form these dense plaques within cells and between the cells. And here's exactly what they look like. This dense plaque at high power on the electron micrograph almost looks identical to the cartoon. Here is a plaque between the cells. These would be these desmogliins, desmocolins binding onto the cells. But the membranes don't fuse, because here's the membranes from one cell to another. The membranes don't fuse. And here showing the plaque binding onto intermediate filament keratin proteins. In the lower power micrograph, here you see the intermediate filament proteins coming in. Look at the space between the membranes. The space between the membranes, but you get a sense that there are integral membrane proteins from one membrane to another membrane that are actually binding to each other, as suggested in that cartoon. Desmosomes are binding cell to cell. They're binding on the lateral membrane domains, and here we see some desmosomes, lateral membrane domains, and notice they're interacting with various cytoskeletal proteins. These happen to be keratin. So desmosome binding cell to cell and binding the cytoskeletons. Contrast that to hemi-desmosome. It is a structure just on the basal domain of the cell, and it's binding onto extracellular matrix components in the basal lamina. So it's binding these basal lamina components through these integral membrane proteins to cytoskeletal components in the cell. These hemi-desmosomes could almost take the name hemi for half. So hemi-desmosomes is like a half desmosome. Hemi-desmosomes bind cell to extracellular matrix on the basal domain of epithelial cells. As we said, desmosomes bind cell to cell on the lateral domains of membranes between adjacent epithelial cells. Now, focal adhesions, which are not shown on this cartoon, create a dynamic link between the cytoskeleton and the extracellular matrix on the basal domain. And we'll talk about those right now, basically. What we're looking at is cellular and extracellular components that provide attachment sites between epithelial cells and the underlying connective tissue. And this is a fairly high power electron micrograph from human skin. Here are the cells, and this is the extracellular matrix, and the micrograph looks remarkably like the diagram. The point I want to make, the hemi-desmosomes, which would be structures shown here, form a very static and fixed attachment site because the hemi-desmosomes interact with intermediate filament proteins. This is keratin. The focal adhesions are forming a more dynamic interaction. Here we're showing, once again, the hemi-desmosome binding onto collagen, the integrin having a receptor for collagen. The intermediate filament proteins are keratin, a very stable or static structure as opposed to a very dynamic structure, the focal adhesions. The integral membrane protein here having a binding site for fibronectin, so a very dynamic structure. The cytoskeletal filaments here are actin. These are microfilaments as opposed to the intermediate keratin filaments in the hemi-desmosome. A clinical correlate that relates to autoimmune disorders turns out that some individuals can produce autoantibodies to various desmosomal proteins, and you might imagine that can compromise the integrity of the skin, of the epithelium, of the epidermis. And there's one such condition. It's called pemphigus vulgaris. This is a condition where cell junctions between epithelial cells in the epidermis are disrupted. You get a lot of blistering, you get a lot of oozing of fluids, loss of fluids, and leads to a lot of cell death. And in the extreme cases, the individuals can actually die. Gap junctions or communicating junctions are junctional complexes between plasma membranes. These form actual channels from one cell to another. They are composed of patches of transmembrane proteins called connexons. So this is a connexon in one cell. This is a connexon in another cell. Connexons are made up of a hexameric group of proteins called connexins. These connexin proteins have a relatively high turnover rate, and they're influenced by both pH and calcium concentration. These connexins form the connexons, and they line up with similar structures on adjacent cells, so they actually will form patent channels from one cell to another, which will allow electrical coupling from one cell to another. It will allow chemical coupling because molecules smaller than about 1,000 Daltons will freely pass. Small ions can freely pass from one cell to another through these connexons, as can molecules like ATP and cyclic AMP. So these connexons allow cells to be in metabolic synchrony with each other. And this is a freeze-fracture electron micrograph of the connexons. And it kind of shows these pores. And then here in transmission view is a very large patch of these connexons. That might be represented here. And this might be a much smaller patch of these connexons represented here. Here's the space between the membranes. I want to point out here, the membranes themselves are not actually fused, but what's lining up one cell membrane to another are these connexon channels. And that's why you get the impression like this on the micrograph. Connexins can be formed from identical or different connexin proteins even within the same cell, and this is thought to relate to the coupling properties of cells. So here you can see a homomeric channel or a heteromeric channel. Here you can see a homotypic connexon one to the other, or heterotypic connexons one to the other. And here showing how the gap junctions might be opened or closed. They might be opened under normal calcium concentrations or normal pH. They might close if there's high concentrations of calcium or if there's an increase in pH. Another clinical correlate here, connexin gene mutations appear to have multiple effects. For example, there's a type of deafness called non-syndromic deafness, not associated with any other signs or symptoms, appears to be an autosomal recessive disorder. There are mutations in 2 different connexin genes that can cause this type of deafness. Mutations in connexin genes might cause various skin lesions. They can also cause dysfunctional migration of cardiac neural crest cells, and this can have significant consequences for pulmonary outflow as the embryos are developing.